For hundreds of thousands of years, human survival depended on a singular, relentless drive, the ability to store fat. Today, that biological programming is surrounded by an engineered environment of calorie-dense foods that actively exploit our metabolic receptors. Our ancestors survived by anticipating severe caloric scarcity. In that context, over-consuming available energy was a necessary physiological feature to outlast winter or famine. This split-screen timeline illustrates the mismatch. On the left, we see the flat horizontal line of historical caloric availability. On the right, the curve steepens exponentially, representing the sudden influx of modern processed foods. The resulting obesity epidemic is the expected biological response to an environment engineered for overconsumption. It is a predictable metabolic outcome rather than a failure of human willpower. When we attempt to reverse this outcome through forced caloric restriction, the body treats the deficit as an immediate threat to survival. Look at this graph tracking a traditional diet. As body weight drops, we see a precipitous fall in the resting metabolic rate, while stress hormones and neuropeptide Y, or NPY, show sharp spikes in the opposite direction. Elevated levels of NPY and cortisol combine to drive relentless hunger, while triggering the maturation of new fat cells to store whatever energy is consumed. Conscious willpower cannot suppress these localized survival mechanisms indefinitely. Eventually, the hypothalamic drive overpowers the psychological intent to restrict calories. Semaglutide steps in precisely where willpower fails. It acts as a total neuroendocrine override, forcing a biological truce between environmental abundance and the hypothalamic drive. This class of medication provides a medicalized pharmacological rewiring of human appetite at the neurological level. When we eat, our intestines naturally produce a hormone called GLP-1 to signal fullness. But endogenous GLP-1 has a half-life of only a few minutes before enzymes break it down. A weekly synthetic GLP-1 agonist injection bypasses this rapid decay, introducing a stable pharmaceutical analog directly into the subcutaneous tissue. The synthetic molecule structure makes it resistant to enzymatic breakdown, allowing it to maintain a sustained chemical grip on GLP-1 receptors throughout the body. Its first major peripheral effect occurs in the digestive tract. The drug significantly slows gastric emptying, delaying the rate at which the stomach releases its contents. By keeping food in the stomach longer, the medication creates a persistent mechanical sensation of physical fullness in the gut. The drug effectively bypasses transient digestive signals to establish a sustained communication channel between the gut and the central nervous system. The true power of this medication lies beyond the stomach. The synthetic hormone crosses the blood-brain barrier, shifting its focus to the central nervous system. It travels deep into the hypothalamus, targeting a specific cluster of neurons known as the arcuate nucleus. Within the arcuate nucleus, the GLP-1 agonist binds to receptors, inhibiting the firing sequence of AGRP neurons. AGRP neurons serve as the biological circuitry responsible for initiating the search for food. By silencing them, the medication effectively shuts down the body's internal starvation alarm. Suppressing these specific neurons forces the brain to lower the body's defended weight set point, allowing fat stores to deplete without triggering metabolic panic. This specific neural intervention disables the evolutionary starvation response at its source. The chemical rewiring also extends to the ventral tegmental area, or VTA, the region of the brain controlling the hedonic reward pathway. Normally, consuming calorie-dense, high glycemic foods triggers a powerful anticipation reward loop, flooding the brain with dopamine and reinforcing the desire to eat. GLP-1 agonism alters this pathway. By binding to receptors in the VTA, the drug physically suppresses the volume of the dopamine spike associated with eating. This suppression severs the link between eating and reward, effectively stripping processed foods of their dopaminergic payload. Psychologically, patients experience this as the cessation of food noise, the constant intrusive background thoughts about the next meal. Because this mesolimbic suppression is broad, patients often experience a blunting of other impulsive reward-seeking behaviors, such as alcohol consumption or compulsive spending. The drug chemically decouples the brain from the pleasure of caloric consumption. However, forced satiation introduces a severe physiological risk. Operating in a prolonged caloric deficit forces the body to pull energy from its own tissues. When artificial satiation signals fail to drive adequate daily protein intake, the body begins catabolizing its lean muscle mass. To maintain essential organ function, the system pulls required amino acids directly from skeletal muscle. To override this fasting state protein breakdown, patients must implement a specific nutritional countermeasure to protect their lean mass. This cellular diagram demonstrates the required biological trigger, reaching a precise threshold of 2.5 grams of leucine per meal. This payload activates the mTOR enzyme, which initiates the process of muscle protein synthesis. Preventing detrimental cellular degradation requires targeted amino acid intervention to maintain muscle protein synthesis, despite a systemic caloric deficit. Preserving muscle mass highlights the broader biological reality of utilizing current GLP-1 formulations. This matrix compares the sustained flatline of lifelong pharmacological maintenance against a violently spiking curve of rapid weight rebound upon withdrawal. If the receptor hijack ceases, the previously suppressed AGRP neurons and dopamine receptors violently overcompensate for the absence of the drug. The brain's baseline metabolic programming immediately re-engages, rapidly initiating a massive caloric drive to reclaim the lost mass. Human appetite and reward behavior now function as adjustable chemical settings, requiring permanent biological management to remain stable.